Okay. First tute. Uh, have you done some of the question from the first tute?
Uh, the tutor says in the first time. Yeah.
First time one, yeah. Because we are still doing the tute of the first tute.
Yes, some questions I didn't have tutor.
I think what is left, this question, and this question. Three questions left now. Because we did other questions. These three questions left. At least we try to make one or two questions today. And then we keep on the new lessons. Oh, this is the tetrahybrid cross. So monohybrid, dihybrid, trihybrid, and tetrahybrid. Four characters take into considerations. So they're all assuming independent assortment of these four genes. What are the probabilities that F2 offspring will have the following genotypes? So it seems the cross is capital A, simple A like this. So it's quite a huge Punnett square we should get for this cross if you want to calculate the probabilities. So we use the shortcut, the method we learn to calculate the probabilities of this A, B, C, D, and E. Have you done this one or other one? This one. Which one you did the left questions? We have three questions to finish.
I tried the second one, sir.
This
The
one.
yeah.
Okay. So then let's do this one and we discuss next time again. I mean, it's like a try. You try to solve it. Take time and try to do it. Um, we try to finally finish all the questions because keep on doing weekly basis, refresh the methods and everything to be also better. Okay, if you take this cross, what is the base for this one? We should have a homozygous and we should get heterozygous. And the next one is homozygous and heterozygous. We could also write the other way, uh, that capital A, simple A, simple A, simple A, right? If we take this is our base for this probability calculation, for this case, it is always resulted genotype is probability is 1. So if we take this one, the probabilities are capital A is this, capital A, simple A is this, simple A, again this. So if we take this one, then capital A, we should get capital A and capital A simple A should be half and half. So is this correct? Do you understand this?
Yes, sir, got it. Yes.
So we have to apply this to get the probabilities now. For example, this one. So this one is similar to this
One,
situation. First one, right? So it's a 1, and the second one is
one,
1, and third one is also 1, right? Yeah. So its probability is 1. And then we take the next one. This is a homozygous dominant, and this one is a heterozygous. Okay, so similar to this, right? Condition A, B, C, and D, similar to C. So in that case, getting capital A, capital A.
2 over half.
This should be half, right?
Yeah.
And then heterozygous and heterozygous conditions. Heterozygous, heterozygous. Heterozygous is this one. So getting this is
1/4.
quarter, and then capital C, simple C, capital C, capital C, simple C. Again this, right?
Yeah.
This one. So A, B, C. Heterozygous, heterozygous, getting heterozygous. Half.
2 half.
Heterozygous, heterozygous, again getting heterozygous, half. Heterozygous, heterozygous, getting half. 108 D. This is homozygous, homozygous, should be 1. Heterozygous, heterozygous,
Half.
half. And then homozygous, dominant homozygous should be 1, right? Half. Yes, I think we are heading to a little bit difficult level in the next coming up lessons, like questions. So after that, we can move. Once we finish this one, we can move to the past papers tuned. That's the second tune we are going to do based on the past papers. For that, we need to finish non-Mendelian genetics. When we finish non-Mendelian genetics, we can start second tune, right? So today, the new lesson is multiple alleles. Multiple allele is the non-Mendelian genetic, and this word multiple allele is the standard the type of non-Mendelian genetics. Polyallelism can be used. It's also okay, polyallelism, but there is another concept coming, polygenic inheritance, polygene. So that little bit of poly and coming polyallelism, someone can interpret it same as polygenic inheritance. That's why most of the textbook, they clearly use only multiple allele. The second concept is polygenic inheritance. So resource book, they use polyallelism and polygenic inheritance. So we also keep this in mind. Okay, both they use. So polyallelism equal to multiple allele. So multiple allele is standard term. Okay, how we understand this? Now, the Mendel genetics, what we have learned, if there is a homologous chromosomes, and these homologous chromosomes, there is a one position here, locus, and there's another locus here. There are only 2, and we mostly try to learn if there is a dominant one is in front, there should be dominant or recessive one is and the second one, these are the two alleles. So in this case, we can clearly say what we learned is two alleles. Now in multiple alleles, so 2 is replaced with multiple alleles. Right. This word multiple replaced here, 2 is replaced with multiple. That means we can interpret multiple is more than 2. The 2 is a standard because it always homologous combination. There are 2 alleles. Now the 2 replaced with multiple word. Multiple word, we can interpret more than 2. Now in this case, if I write like this is a one allele. This is another allele. This is another allele. Right? So if I write X, Y, Z, that is quite fine because it is more than 2. Now this X, Y, Z can interpret this is more than 2. In multiple allele, we are going to learn how can X, Y, and Z. So these all three going to occupy at the same locus. Simply, there are 2 chairs, but now we have 3 people. So if there are 2 chairs, there are 3 people to sit on 2 chairs. How could we do that? Now, how can we understand much better? So let's assume there is a chromosome in human human cell, 23 pairs. So if there are 23 pairs, there is one chromosome. Example, let's say chromosome number 6. 1 to 23, chromosome number 6 is this. And from chromosome number 6, there is a one place allocated for a particular character. And as we learn, there are only 2 alleles can be existed. Because these homologous chromosomes allow only 2 alleles to place in this position. Here one and here one. It could be a dominant, homozygous dominant, heterozygous, or homozygous recessive. Any of the combination allowed to come. Now, in human population, if we take as a whole population, there are 3 alleles which prefer to stay in this position. If we take a whole human population, there are 3 alleles existing. In this case, we can still keep the same rule. No, only 2 can come at a time. We allow only 2. Let's say this person, his name is A. Oh, we take another letter. His name is, yes, capital A, his name. And now this person can take only 2 alleles. So he can take X or Y. And there's another person existing, is this B. Person B can take X and Z. Person C can take Y and Z. So something like this. Or maybe even person D is there, he has X and X. Person E is there, he can take Z and Z. So it is distributed among every person, only 2 alleles, but in different combination. So this is called multiple alleles. There are several alleles among human population, and these alleles distributed always two alleles. As a result, people might have different combinations. So based on the combination, they can express the phenotype. For example, X can express a phenotype, Z can express a phenotype, Y can express a phenotype. Based on their combination, which one is dominant, they can express. Or as we learn incomplete dominance, if one is trying to express, other one also try to give some effort, mix will come. So something like that we can understand. So in short, several alleles existing in the population, but only two alleles occupy at the loci. As a result, people are existing with different combination of these three alleles. This is multiple allele. Did you understand this?
So eye color is multiple alleles.
Uh, eye color is multiple alleles. Eye color is not exactly multiple alleles, but we take always one example. Almost all textbook they take one example for this. That is the most understandable example and easy to understand. Having said that, it is not only multiple alleles. So there are many multiple alleles existing in different places. Uh, Most of the time, color is uh, coming from polygenic inheritance. But when you take the hair color, eye color, skin color, we have to look into different level what kind of concept existing for hair color, eye color, and uh, the skin color. So uh, eye color is multiple alleles. So we have to look how many eye colors and how many alleles. So for that, we'll keep it. We park that one to check whether what are the other types of multiple alleles in human genetics. So we take the right example to understand this human blood groups. So you have done circulation, right?
Circulatory system.
Circulation.
Uh, Circulatory system is
Yeah.
yes, slide school.
Okay. Right. So I mean, unit 5, we learn actually human blood groups. Now we are in unit 6. There's no need to explain human blood groups as we learn from unit 5, how a human blood group decide and what are the existing blood groups. I mean, if you have uh, not much clear about the human blood group, briefly, it is like this. If you take a red blood cell, so this red blood cell, that's a cell membrane, let's say this one, it has a protein on the membrane. So this is the proteins. And then protein is designated as protein A. And there is another uh, protein existing in red blood cells. This protein is called protein B. So this is a red blood cell. This is a red blood cell. When we finish unit 5, we also finish immunity. So that means you have a good knowledge of immunity, you have a good knowledge of a circulatory system. So to understand the human blood groups, we need both immunity and circulatory system. Now, person having this kind of a red blood cells with the protein A and person with the protein B has in the plasma they have antibodies. This is the antibody for uh, protein B. And these people, they have antibody protein A. So plasma has antibody for protein A and B. Now, what we have to understand from immunity, antibodies can bind with antigen because antibody has a binding site for antigen. And after that, what is the importance of binding? What happened next? Everything will be explained in immunology, immune system. At the moment, we just keep antibody can bind to the antigen. So in this case, this person A antigen cannot bind with this antibody B. In this case, person B antigen cannot bind with the antibody A. So there is no reaction. And there's no reaction here. So there is no reaction at all. But what happened if the blood donate A to B, or B to A, whatever. Now this red blood cell is going to the other side. So I draw this one in different color maybe. Now after moving to person B, this A antigen is here. That's A. And A antigen is here now. Now this is a chance for antibody to bind with A. This antibody bind with antigen A. The binding mean there is a reaction. There is a event after that take place. This event is not favorable event because always antibody binding mean the immune system is reacting. Then immune system is reacting mean it try to get rid of these kind of thing out of the body. The A red blood cell is not suitable for the B person immune system, it try to remove. So we have, I mean, I assume that we haven't learned immune system and circulatory system in much. I just try to explain there is an incompatibility in people about the different blood groups. So we cannot donate blood from one person to another person without checking the blood group. Now, we'll keep this a little bit away now because we anyway going to learn blood groups and immune system. Now you can understand, if the A is a protein, this protein code is in the DNA, in the genes. So there is a gene for protein A, and there is a gene for protein B. So this gives us some kind of idea. The gene is responsible to make the proteins. So then if we take human population, how many alleles existing for blood groups? So we can write like this. This is a one allele, and this is another allele, and this is another allele. So A, B, and O. So A, B, and O are the alleles for blood groups. Now this is fitting to the concept what we learned, X, Y, Z. There are three alleles, X, Y, Z, existing in human population. So there are three alleles, A, B, O. And we write usually these the alleles I superscript A, I superscript B, I superscript O. Now in this IA, IB, IO alleles, these two are dominant. And this one is a recessive. Dominant and recessive. That means when O is with A or B allele, only A expressed. So what are the combinations now we can write? We can write one is I, A, I, A. I, A, I, O. So this case, A is dominant, A is expressed. Then the blood group is A. And the second one, IB, 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 IO. This case, B is dominant, blood group is B. If we get this combination, it is recessive, homozygous recessive. And the person has O blood group. Do you see anything missing? Any combinations?
No, sir.
Yes. What about
Ah,
this?
yes.
So now, if IA allele comes, IB alleles come, we have a question. What is the phenotype? So, because we learn dominant express. Now we have a very special situation. Both are dominant.
Incomplete dominance.
We can't say incomplete dominance, because incomplete dominance is uh, dominant and recessive. It's like one is dominant, other one is recessive. But here both are dominant. It's close to incomplete dominance, but we have to little bit distinguish between incomplete dominance. That's why sometimes this incomplete dominance is a little bit mixed up with another concept. Now, incomplete dominance, what we said, the flower color is red, and the other flower color is white. And if they mixed, we get a pink. The flower color is pink. I can't write. Pink color is not here. Anyway, just like this, pink, red, white, pink. This is incomplete dominance. Now we take the red blood cell story. What we learned, red blood cell, the blood cell is here. There is a protein express, that is protein is A, the other protein is B. Now, what if I write the protein A is here, protein B is here. Both are. A are here, A and B. It's not a mixture of proteins. So incomplete dominance, we should expect a mix, blending of these two proteins. But it doesn't happen. A separately, independently make A, B also make independently B. Now we can learn two concepts. One of the two concepts with one example, multiple alleles are more than two alleles occupying the same locus, right? So that's important. If we want to define these multiple alleles, we can define it more than two alleles occupy the same loci. So that's very important for the definition, same loci. The loci is this position. There's no other place, only two seats allocated. They can't go and sit other places. Like for example, if we have a three, okay, one sit here, other one is going to this place. No, it's not possible. Only the two seats, it should be sit on the same seat. So that's why occupy the same loci. So that is multiple allele concept. More than two alleles, so many alleles occupy at the same loci. That's a multiple allele. But now we have again a very different situation. In this situation, we can't explain in incomplete dominance, because incomplete dominance is a blending of these two phenotypes. But here it's not blending, they independently make their phenotypes. Both phenotype express. So this is defined as both expressed. One example, two concept. The concept one is a multiple allele. This example is called codominance. That's called codominance, right? Codominance.
So what is the most abundant blood group, sir?
Mostly A and B